Hello, everyone. Good evening, and welcome to the third session of CAPS Talk series. So, last time we have dived deep into the sea of protein domain. So, today we'll be taking you to the journey over olfactory cues. So, over to you, ma'am.
Thank you, Shailya. Today we will talk about chemosensory reception, as it were. So we will start with something primitive and relatively easy to understand, such as bacterial genomes. Okay, and here bacteria are constantly having to survive in the midst of other bacteria, not only of their same kind but of other kinds. And some could be pathogenic, some could be non-pathogenic. And in this process, the chemicals are coming out. They're spewed out by the bacteria, which are shown in this schematic as blue circles. And then these are being perceived by other bacteria, let us say of other species. And as soon as this chemical concentration acquires a certain minimum threshold, these bacteria shown in oval shape is going to not just perceive the presence of these chemicals, but begin to do something, which is what comes under the category of quorum sensing. And therefore, it will sense the concentrations of these small molecules and start to begin some protein production in a series of proteins getting activated and other genes getting translated, expressed, and so on. And today we are going to discuss about just one of these proteins, not the ones that actually respond to the concentration of these small molecules, but those that are actually involved in the synthesis of the small molecules. And one of them is called as Luxes. And Luxes, the crystal structure has been determined in back in 2001. And what we have tried to do is to follow the evolutionary track of the Luxes over many bacterial genomes. And for this, two of my lab members in the last few years ago, Rajas Rao and accompanied by Nasir Pasha, had actually tried to follow the genome-wide survey of Luxes in different bacterial species and try to compare. And what I found was really quite fascinating. So I would, at this point, request Rajas to explain his journey in the Luxes computing. Rajas, please.
Thank you, ma'am, for giving the opportunity to present the work. It has been a great honor and privilege to work at the CAPS lab, to be part of the legacy of the CAPS lab. So I'll just share my slides. I hope it is visible now.
Yes.
Okay.
Please go on.
Thank you, ma'am. As ma'am just talked about now, different bacteria in an ecosystem communicate via a mechanism called as quorum sensing. So there are two different, as ma'am also talked about, different mechanisms of quorum sensing. There are two types of quorum sensing, where one is called as auto-inducer 1 mediated and the other is auto-inducer 2 mediated. So the diverse species of bacteria in an ecosystem communicate via auto-inducer 2 mediated pathway. So this as to the biosynthesis of the molecules involved in this pathway involves different enzymes. One of the enzymes is the Luxes, which catalyzes the hydrolysis of a metabolite called as S-ribosyl homocysteine into auto-inducer 2. So we hypothesize that since diverse bacterial species use the same quorum sensing molecule to communicate among themselves, the proteins that are involved in biosynthesis of this enzyme must have evolved early, and they must have strong residue conservation. So with this hypothesis in mind, we studied evolutionary divergence of Luxes through phylogenetic as well as structural perspective. So we did various sorts of experiments to trace the Luxes divergence. As ma'am mentioned, we performed phylogenetic analysis on large number of Luxes homologs. In fact, more than 3,000 Luxes homologs. On the selected Luxes sequences, we also performed analysis of conserved motifs that are motifs that are conserved among all different Luxus sequences using an in-house tool developed by the CAPS lab called as Motif S program. And we also try to understand Luxus divergence from structural perspective by performing modeling of different Luxus homologs. We also perform electrostatic analysis of the conserved regions of Luxus homologs, and we also try to understand how the divergence of Luxus occurred from a structural perspective as well. From the phylogenetic analysis, we found that Luxus evolved early. In fact, it formed 5 highly conserved clusters. And interesting thing we observed among these clusters were habitat specific, meaning in different clusters were populated with specific with bacteria that were localized to a specific habitat. For instance, one of the cluster had a large population of gut-residing bacteria. The other cluster had a large population of extremophiles. The other population had a lot of plant or food-associated bacteria, and the one more cluster had mostly gram-negative pathogens. So this was something quite interesting, which we inferred that since the compound involved in interspecies communication is the same, and the divergence must have occurred quite early, based on the fact that we are seeing five clusters that have diverged together. We inferred that this might be a case of conserved evolution, where the Luxus structurally, depending on the type of habitat, must have undergone some sort of convergent evolution so that they, in such a way that Luxus homologs from in the same habitat are more similar to each other. And when we looked at certain conserved motifs among the representatives of different clusters, we found certain motifs that are conserved across all the 5 clusters. And at the same time, we also found certain motifs that are unique to a certain cluster. They were quite well conserved in one particular cluster, but these motifs were not present in other clusters. And when we tried to look into the structures of Luxus, we found that the protein across all the homologs has one particular strongly conserved catalytic site, which is well exposed and which is important for catalysis of the S-ribosyl homocysteine. But when we modeled representatives, when we constructed 3D models of representatives of each cluster of Luxus sequences, we found that while there is a large amount of conservation, we also found that many regions also have also diverged structurally. They were structurally quite different, which also include certain significant regions, such as one particular regions between the residues 125 to 131, which acts as a gate to active site. So we inferred that such sort of divergences could also contribute to the role of Luxus, which enables it to adapt to different sorts of bacterial environment, while at the same time evolve a mechanism to have a common communication mechanism between different sorts of bacteria. With that, I would like to thank again for giving me the opportunity to present these results.
That was great. Thank you. Thank you for the very crisp but interesting story of looking at bacterial Luxus proteins. Looking back at primitive bacteria is one way to start, but that was one story. But let us now try to step back in time, all right, in which we would ask questions such as, what were we learning just as the genome sequencing project had begun back as early as 1999? And for this, let us look at some of the literature that had started accumulating over time. And in 1999, for instance, this was the time when the chromosomes for individual species like the yeast and all had just been sequenced using shotgun sequencing approach. And for instance, looking at the data in front of them, many bioinformaticians all over the world, like Chris Sander, David Eisenberg, and so on. And lots of them had already started to analyze. And one of the things that I've just picked up as an example, and this is going to be like at least 2 dozen papers around that time, but this paper that had appeared in this journal called Bioinformatics says structures are required. Until the 3D structures of these orphans are experimentally determined, more sensitive bioinformatics tools will aid in placing genomic orphans into their proper protein superfamilies. What I mean to say is that these are can be viewed as open reading frames, but these are also orphans because they don't easily connect with pre-existing protein families. All right, so this was the learning that was coming up, and also because we know that nearly 40% of genomes are made up of membrane proteins. So how do membrane proteins in general fare? And how are we doing with respect to structure prediction? And for this, I would like to share this graph, which shows starting from the 1960s, as the globular protein structures got determined 18 years away, how the trend was actually predicted and found to be correct. On the other hand, 1985, when the membrane protein structures like the rhodopsin got determined, whose structures got determined, let us say 18 years away, we are doing very well with similar trends. And it was predicted by 2005, we will have about 100 membrane proteins. So in reality, even now, the total number of proteins, membrane proteins whose structures are available is really a small number. And that kind of warrants for the fact that we ought to, as bioinformaticians, look into membrane proteins that are coming from genome sequencing projects. And to kind of motivate this, already people have been publishing. And in 2003, for instance, this is another group which had looked into a few bacterial genomes and used bioinformatics algorithms to tease out the membrane proteins of their interest. And this was going on. And we were also reading up papers such as genome sequencing analysis. And at the time, there was this paper published in Genome Biology where they had looked at the classic membrane proteins, and one of the most attentive, that membrane protein which acquires maximum attention, which are the G protein coupled receptors, because they are drug targets. And within the human genome, this paper had described the way by which, using simple phylogenetic algorithms, one can infer functions of orphan GPCRs just because of the co-clustering of known GPCRs, meaning GPCRs with known ligands with respect to those which are orphans. And this is like a guilt by association. And I kind of waved this paper in front of Raghu Prasad Mejpali, who was my third PhD student back in 2002. And he immediately started off to get interested. And he had been looking at GPCRs in the Drosophila genome then. And then I asked him whether we should try and co-align, combine the human GPCRs with the Drosophila GPCRs and see if we are able to likewise infer function through phylogenetic principles. And the next 3 or 4 years, it got Raghu Prasad busy because first of all, we are dealing with 500 sort of protein sequences, and these are all membrane proteins, and the alignment becomes very hard. And these were also the times when people had not started using rapid phylogenetic or alignment techniques such as muscle or mega so much in practice. And there we were sitting with kind of several A4 sheets of the phylogeny that he had got with 500 GPCRs, both from human and Drosophila. And then he had just pasted them up and we had put it on the floor. And he was crawling on the floor, over the floor on the phylogeny. But I was standing next to him with a laptop and reading out accession codes. And what we were trying to do is to understand which cluster are we staring at. And this took us a great deal of time because it's not only getting to know the phylogeny, because then we have to infer more things. And this gave rise to a whole lot of time. And we then decided to concentrate on specific clusters and infer what we will. And one of the examples is shown here, but we were able to infer on domain architecture, cellular localization, the possible endogenous ligand, and so on. And here you can see that there is an orphan Drosophila GPCR. And because of the phylogenetic association, we infer that it may be a arachidonic acid receptor. And there have been attempts to actually take this up forward. And you can see that nearly 45 papers had come following up on our prediction of that one prediction, but we actually made nearly 60 predictions. And you can see as we look at in the flyways, as of now, this particular entry has already been accepted as a probable GPCR. And it is quite encouraging also to note that nowadays, as of 2021, Flybase is adopting this strategy now, what we had proposed many years ago in 2007. Computation of Drosophila melanogaster genes relevant to disease based on their orthology to human disease genes. That shows the power of being able to extrapolate from one genome to another, even though it may be from human to fly or fly to human and so on. All right. So this was encouraging, but that is just a prelude to our second story on the GPCRs. And whatever we had learned with GPCRs was useful because then Raghu Prasad had already started picking up olfactory receptors and we saw that the GPCRs in Drosophila would not cluster with the olfactory receptors in the same genome. And they are just existing as distinct clusters. And the topology of the olfactory receptors is also inverted with the N terminal on the inside facing the intracellular milieu and so on. And this was the same time when Carlson's group had started to map the different olfactory receptors in the Drosophila antenna. And armed with all this excitement, we proposed and submitted a grant with few labs in the Pikui, who is in the Riken campus in Tokyo. And together, we started to work on olfactory receptors in few different model organisms. And this is going to be presented by Naga in our group. We had to make sure that the alignments of different GPCRs, whether it is within genome or across genomes, are going to be sound and reliable. And Naga is going to briefly narrate this as a part of our second story. Please.
It's my pleasure to join for the CAPS talks. And I have a long time association with Professor and Lab 25 from the year 2008. Olfactory receptors are belonging to membrane protein, particularly class A type of membrane protein. They have attracted for Nobel Prize in the year 1991 by Linda, Bug and Axel. Still, the practical implication of olfactory receptors are remote. One could broadly contemplate to utilize olfactory receptors for aromatherapy, pharmacogenomics, and even personalized medicine. To reach these heights, primarily, it is very important and necessary to understand the sequence properties and to identify right drug to the right receptor. And also to prove the slogan, let you smell the drug and get cured in inhalation therapy, sequence analysis definitely play a major role, and it is the starting point in olfactory research. As I mentioned, Linda and Bach clearly explained the olfactory signaling pathways and also the topology. Here, human olfactory receptor retains its topology as N out and C in topology. Olfactory receptors in human having 7 transmembrane helices connecting 3 intracellular loop and 3 extracellular loop outside. We want to do the exercise for human olfactory receptor. From the lesson 1, we have understood when we give a chance of clustering human olfactory receptors of 371 candidate receptors, they have given 10 distinct clusters. Here, we could clearly see the distribution of 10 distinct clusters, and we named clockwise HSC1, C2, C3, and so on. Notably, cluster HSC1 remains distinct, and it is not co-clustering with other human olfactory receptor. In the lesson 2, we wanted to perform cross-genome phylogenetic analysis of human olfactory receptors with candidates from Drosophila olfactory receptor. In this cross-genome phylogeny, Drosophila olfactory receptors are not co-clustered with human olfactory receptor. It is due to the reverse topology of the Drosophila olfactory receptors. As I mentioned, the topology makes the difference. N out and C in topology retain in the human olfactory receptor, whereas N in C out topology is from the Drosophila olfactory receptor. That's how it is not co-clustering with each other. With this learning, we wanted to perform cross-genome phylogenetic analysis of human olfactory receptor with few fish olfactory receptors. Fish's olfactory receptors are very co-clustering with HSC1 in the human phylogeny. Here, it is not co-clustering with the other human olfactory receptor. In the literature, it suggests that it could be because of the fish olfactory receptors are sensing waterborne odors. So the HSC1 belonging to sensing waterborne odor. When we want to perform the other exercise with amphibian olfactory receptor, we have done cross-genome phylogenetic analysis with the fishes and amphibian olfactory receptors. We found the frog olfactory receptors are co-clustering with already established human olfactory receptors in HSC1 and with all other human olfactory receptors. This clearly emphasizes that this association of class 1 type belonging to sense the waterborne odor, but class 2 type are helpful in sensing airborne odors. This depicts that the dual habitat of the amphibian, the frog. With this understanding, we wanted to have a tool called TM motif, and which is a very helpful tool to view the aligned set of olfactory receptor in the WebGR coloring scheme. And the conserved residues in the mouse over, we could identify the conserved residue and also the substituting residues. This way, TM motif helpful to understand in the multiple set of olfactory receptors, one could easily find out the conserved residues and also the substituting residues. We have deposited the sequence information such as topology, cross-genome phylogeny, representative of olfactory sequences and its respective secondary structure, ligand binding sites with the conserved residues of 5 model organisms in the DOR database, the database of olfactory receptor. The database of olfactory receptor having a very fantastic features, having the sequence and structure information, where it includes the information of Homo sapien, Mus musculus, Drosophila melanogaster, C. elegans, and yeast. And we could able to get its alignment and phylogeny for the unigenome and the cross genome, and also the conserved motifs, predicted topology, secondary structures, and the ligand binding sites. With this, the DOR output files can be used for docking studies to screen bioactive compounds from the medicinal herbs. Also, appropriate ligand receptor pairs can be further validated by in vitro and in vivo analysis, ultimately to find effective drug to known target olfactory receptor for the various diseases such as asthma, COPD, lung cancer, and etc. Information about olfactory receptor of model organism, knowledge and homology within and across genome saves wet lab time and helps to improve the observation of olfactory receptor signaling pathway and disease regulation in aromatherapy. With this, I would like to thank Professor and team for making the door open to olfactory science in silico in India.
That was very nice, Naga, for telling us that these aquatic ORs could be a specific cluster and so on, and also emphasizing that the transmembrane helices are important. But where does it take us next? So in fact, the transmembrane helices are so important, and I gently pointed out to the team that although the prediction of transmembrane helices is pretty accurate, more than 90%, there could be 7 methods, and earlier we had found that the 7 of them do not particularly agree on the boundaries of the transmembrane helices. And then the next member in this team who joined the lab, Snehal Kape, went ahead and looked at 20 different prediction algorithms and said, yes, they do not particularly agree. And she went on to create a consensus, which she will tell later on. But as this was going on and Snehal was explaining her contribution, on the care for transmembrane helix predictions in NCPS in a center-wide contribution for the students in an annual work seminar. Dr. Axel Brockman, my colleague, listened to it and came forward to us and asked, can we now look into honeybee ORs where they are having some trouble? And this is the beauty of in being in NCPS, it's highly collaborative, so they happen very naturally. And then we definitely went ahead and worked with him, and we realized that it's not only the accuracy of getting to know how many members are there in a particular genome, but also how accurate it is, but also about the numbers. And in the case of olfactory receptors, although the ORs do not particularly co-cluster with the GPCRs, and therefore one is not going to have crosstalks when we do sequence searches. In general, ORs have low sequence identity. Plus, they do not have functional motifs like the GPCRs, such as the DRY or NPX excited motifs, making ascertaining the boundaries more difficult for ORs. Plus, the ORs have long intracellular loop regions, and one transmembrane helix can respond for the other transmembrane helixes, so we don't know. We are particularly looking at the third or the fourth or fifth transmembrane helix and so on. And finally, ORs do occur in groups. In the chromosomal level means they can occur as gene repeats, act as neighbors. And for all these reasons, there are challenges. And due to all this, there began in our story the genesis of InsectOR webserver, which will be explained by Vikas Tiwari from our lab currently. And this will be followed by a quick narration by Snehal, who had used this approach to compare Asian and European honeybees, and also social and solitary honeybees, and so on. So do wait for your stories.
Hello. Thank you, ma'am. And good afternoon, everyone. As it was mentioned that insect olfactory receptors are a difficult class of protein in terms of finding them or identifying them from a given insect genome sequence. And to address this problem, we had come up with an algorithm which is being presented in the form of a web server called InsectOR. So this is the home page of the insectOR web server, which can be accessed through the given link. And I'll be talking about what it predicts and how it predicts. So in terms of getting the input from the user, we require the genome sequence in FASTA format of the target insect in which we want to predict the olfactory receptors. And along with that, we also need the known olfactory receptors as a query sequence. And for that, we can take the OR sequences of the already annotated insect genomes, for example, from Drosophila or from the mosquito. Once we have those input files, we start by aligning the known query sequences to the given genome sequence, for example, on the virtual scaffold. And by the alignment, we find the best query sequences aligning to each of those subclusters. For example, in this case, we can identify OR1 as the best scoring query sequence for that particular genomic locus. And once we have the best scoring query sequences, we also try to identify the remaining positions where all that particular query sequence is being aligned, although that might not be the best query sequence for all of the genomic locus. So once we have that identified, based on the overlap between the different alignment regions, we stitch them together to build a better gene model. And in similar way, we will have, so here I have shown only for the few representative query sequences being aligned to certain genomic regions, but in that way we'll have multiple query sequences aligned. So we filter them based on the length of predicted protein length, and we select the best query sequence, and we call that as a prediction one hit. Now, once we have the prediction one hit, we want to refine this gene model, and we extend the gene boundaries, and maximum allowed region is like 6,000 base pair, which is like average genic region between the insect genomes. And we use another gene prediction tool called GeneWise to predict the another set of proteins for the same genic region. So, and that we call as P2 hit. Now we have two set of predictions for the same genomic region, P1 and P2 hit. And in cases where they do not overlap, we retain both of them. And wherever there is an overlap, we retain the best predicted gene model. So best in the sense the completeness of the gene model and also the absence of the serogenizing element. finally, we refined the gene models by finding the start and stop codons. And once we have the final gene models, we do several validation and analysis by identifying the signature olfactory receptor motifs, and also by predicting the transmembrane helices. And this web server also allows to search for the user specified motif in all of the predicted hits. So give you as an example of the sample output from this web server. This is an example for the blueberry bee called Hebropoda laboriosa. So we first summarized all the predicted hits. So in this case, like there were 144 olfactory receptors predicted, and then we categorized based on the completeness, presence of the pseudogenizing element, and how many of them have start codon. And further, within 144, not all of them will have the signature motif. So those who have the signature motif has been categorized separately. Subsequently, we also provide the tabular format, which will tell the start and stop position and on which strand the OR gene is present. And as I mentioned, like we are using 3 different transmembrane helix prediction tools, and also we are providing the consensus TMH prediction. And as a bar graph also showing like how many of the predicted ORs have how many number of TMH transmembrane helices. Generally, they should have 7 transmembrane helix, but as ma'am mentioned, these are pretty difficult to predict. So many tools might predict a lesser number of transmembrane helices, even for the properly annotated ORs. We have also integrated a genome browser to visualize the predicted hits on the given genomic locus. Also, if the user has already annotation from other genome annotation pipelines, those can also be visualized and compared. And finally, we are also providing an option to search for the user specified motif in the given or the predicted all the hits. That option can be utilized once if the user already has some information regarding the presence of certain motif other than the signature motif of the olfactory receptors. Further, we also compared this web server with the popular genome annotation pipeline called Maker, and this was specific only for the prediction of the olfactory receptors. So we found better performance than the Maker, which is a generic pipeline for predicting not just the olfactory receptor, but for annotating all the genes. And for further details, the article can be referred, and also we have a GitHub link to download the standalone version of the web server. Thank you, and over to you, ma'am.
I will now request that Sneha's recording be played because she talks about her experience comparing different honeybee ORs.
Hello, I'm Snehal. This is me here in the middle of this picture with Professor Saudamini, my PhD guide, along with Dr. Axel Brockman. While I was doing PhD here in Professor Saudamini's lab, I was involved in 2 collaborative projects with Dr. Brockman's lab. Both of these projects focused on olfactory receptors coming from various bees. And today I will give a brief overview of these projects. Insect olfactory receptors are very interesting, firstly because they are quite diverse. For example, if you see here in this phylogenetic reconstruction of olfactory receptors belonging to various different insect orders, you can see that they form these different clades, which means that if I start with olfactory receptors from a species belonging to Diptera like Drosophila, and I start looking for these olfactory receptors in a species belonging to another order like Hymenoptera, then I may not be able to find all of the olfactory receptors belonging to this particular insect order, which means that it is a very interesting bioinformatics problem, and we were excited to make a tool or form a kind of analysis pipeline which can really help to annotate these olfactory receptors better. So here we started working on olfactory receptors of honeybees first, and first we targeted a species called Apis florea, which is a kind of a dwarf Asian honeybee, and it is related to Apis mellifera, which is the European counterpart. It was known that the morphology of these 2 bees is quite different from each other, and particularly in terms of their antennae, they were quite different. So the question was, do they have same number of olfactory receptors, or they may vary a lot across the 2 species? We also knew about few olfactory receptors that were pheromone receptors from Apis mellifera, and we knew about the pheromone components of the 2 bee species, and that they were very similar to each other. we defined our bioinformatics pipeline. Here we started with olfactory receptor genes and proteins from the other insects and searched into the newly sequenced genome of Apis florea. With this pipeline, we ultimately found 180 olfactory receptors. And when we compared this with what was already known in the genome database at that time, we found that we had 80 completely novel olfactory receptors after our analysis. And the total number of olfactory receptors that we found in Apis florea was now very well corresponding with what was present in Apis mellifera. After this, we performed phylogenetic analysis of all these olfactory receptors and found 22 different clades or subgroups. And then we identified few subgroups and their putative functions. For example, there was one particular clade that had an olfactory receptors from Apis mellifera, which is known to be a queen pheromone receptor. We found its perfect ortholog in Apis florea. Again, we found another clade which we thought could be putative cuticular hydrocarbon receptors as they were highly different across the two species. After this, we performed comparison of the transcriptomes of drones and workers or males and females. And when we did this, few of our putative functions or few of the predictions that we made before based on only the phylogenetic analysis, those could be confirmed with this analysis. For example, the putative pheromone receptor was again highly expressed in drones compared to workers, whereas putative cuticular hydrocarbon receptors were highly expressed in workers. After that, we decided to look into olfactory receptors of solitary bees. At that time, none of the solitary bees were studied in detail for their olfactory receptors. Uh, Previous literature had shown that typically eusocial species or species like bees had higher number of olfactory receptors to other solitary species like Drosophila. However, there was no information available about solitary bees. So do these solitary bees have lesser number of olfactory receptors compared to honeybees? That was the main question. So here we decided to look at the genomes of two solitary species, Habropoda laboriosa and Euphoria novangbe. And when we applied an approach that was similar to what I showed before, we found 112 olfactory receptors from Euphoria, which was quite uh, less compared to other Hymenopteran or other bee species. Whereas in Habropoda, we found 151 olfactory receptors, which was kind of closer to the number that is seen in other bees or other Hymenopteran species. We again performed phylogenetic analysis as before and found some interesting observations. For example, the putative queen pheromone receptors that were present in Apis mellifera and Apis florea, they were almost absent in the solitary bees, as one would expect, because they do not live in colonies or have drones and workers. When we study, when we looked at the correlation between the total number of olfactory receptors and genome size, uh, the olfactory receptors coming from other insect orders than Hymenoptera had quite positive correlation. But the ones that were coming from Hymenoptera, they had a very diverse number of olfactory receptors, and there was no clear correlation in this total number of olfactory receptors with the degree of their eusociality. So we think that the number of olfactory receptors present in Hymenoptera, they are not solely dependent on the degree of eusociality, and other ecological factors might have a role in deciding these numbers as well. In another study, we performed transmembrane helix predictions, and we tried to review performance of various transmembrane helix prediction algorithms with respect to each other. And here we first gathered information of all available transmembrane helix prediction tools, which was more than 20 at that time point. And then we applied these to newly newly identified GPCR structures, and we looked at the accuracy of this prediction. When we did this, we saw that accuracy typically ranged from 82% to around 94%, with some prediction tools performing much better compared to other, like MEMSAT SVM performed much better. Then in the second approach, we compared the predictions from one tool to the predictions from all the other tools. The idea behind this was that if there is one tool that fits well with predictions from most of the other tools, it might be more representative of what the transmembrane helix predictions look like. So when we did this, again we found that few methods performed much better than the other, and MEMSAT SVM was one of them. Such kind of analysis give us an idea about which prediction tools perform better compared to the others in which context, and they can be applied before performing further analysis like prediction of structures of transmembrane helix containing proteins. Thank you.
All right. So I hope you found our story on the olfactory receptors quite interesting, especially the computational angle, which is to use kind of InsectOR webserver to stitch and recognize the genic regions that code for ORs, as well as the choice of multiple TM transmembrane helix prediction algorithms to drive such a recognition. And this was our second story. And just to give you an idea on the time frame, Snehal joined in 2012, and Naga had joined our lab before, and Vikas had joined after. So this is really stretched out over many years of time, and we were happy to collaborate with the people in the process. But to just continue and then take us to the third story, which is on the odorant binding proteins. This was natural next because on one hand, these are biologically connected as well, because you can see that the odorant binding proteins actually initially meet the odorants right at the nasal epithelium, and then they trap the molecules which are otherwise volatile, and then take them all the way up until they find the odor olfactory receptors. And in doing so, they have this sort of a structure which is very conducive for trapping the volatiles. In the mammalian genomes, this is performed by the structure is shown here. And the reason why I have put up this picture of Professor Archunan of Bharathidasan University is because we collaborated a great deal with him. So we had been looking at the mammalian olfactory binding proteins as one of our superfamilies of interest, and and we are looking at alignments of such difficult, distantly related proteins. And therefore, we had started to publish about the odorant binding proteins in mammalian genomes. And this was noticed by Professor Archunan, and he literally met us on the corridor, and then the collaboration began. In NCPS, he had met us in NCPS and then asked whether we can now look at some odorant binding proteins, but in rat and mouse and so on. And that's because he has developed a Center for Excellence on Pheromone Research back in Bharathidasan University, where he belongs. And this was the start of our rather long story on odorant binding proteins, because it did not stop just with the mammalian OBPs, but also with the insect OBPs, whose structures are completely different. These are rich in helices, and these also became interesting to us in general, and also because of our outreach by Dr. Bernard Hoffman, who became then our long-term collaborator as well. But this collaborator is normally, he had been working in the University of La Jolla in La Jolla Islands. And we, in fact, co-shared a student, Malini Manoharan, who can be seen right in the middle in this picture. And Malini will be describing the story of how searching for odorant binding proteins of this nature in mosquito genomes become relevant because in Hawaiian Islands, there was a huge outbreak of chikungunya virus, and that became most Malini's PhD project. So I would like to request Malini to present this third part of this of our story. This is the part of the third story which we are presenting to you. Yeah, Malini, please.
Thank you so much, ma'am, for the introduction. It's always good to be back to what I always call my second home. I had great experience working in NCPS, and I'm very glad that I can talk about my work again. So we had, like ma'am told you, we wanted to characterize the odorant binding proteins in mosquitoes. The major reason was that global warming is happening, and it's happening even now, and this led to a lot of spike in deadly diseases, and also increase in the mosquito population as well. And there were reports from the WHO that said that there are 3.3 million people who are at risk to malaria. So we also wanted to look at different strategies on how we can help in preventing the spread of such infectious diseases. So our strategy at that point of time was to look at the olfactory mechanism of mosquitoes. As you can see in this animation, what attracts the mosquitoes to the humans is the odorant that is present on the human skin. And we wanted to understand how exactly this works in a mosquito. And to just give you a sense of how it happens, how it works. So they have a sensory neuron, and there are dendrites, of course, and then there is something called the syncytium lymph. And if you take a closer look at the syncytium lymph, what you will find is it has a set of proteins called odorant binding proteins. So the previous talks, as you have heard, talks about olfactory receptors. So these are the proteins that actually start the signal, which are present on the neurons. But the syncytium lymph has this very unique proteins called odorant binding proteins, which we understood, and we also realized that it can do a lot of functional differentiation in ways it recognizes different odorant proteins, and we had published a lot on this as well. So what we understood is when an odorant molecule from an external environment enters the syncytium lymph, there is a odorant binding protein to which it binds, and this odorant binding protein then binds to the olfactory receptor, releases the odorant, and this binding actually triggers a signal suggesting that they have identified their host. So what we wanted to do was to we wanted to understand the distribution of odorant binding proteins in different mosquito species. We also wanted to understand the specificities towards substrate binding. And we also wanted to learn how they transport the protein, how the ligand binding happens, how the release of the odorant molecule happens. Today, I will stick to talking about just the distribution of OBPs in different mosquito species, and the other two definitely for some other point of time. So what we ended up was we started off with a limited set of previously annotated odorant binding proteins. And via our sequence search, what we were able to add on to the repertoire is 110 new odorant binding proteins, which were never characterized before. Um, and if you look at the characteristics of these odorant binding proteins, they have a very a nice sequence pattern, which is of 6 conserved cysteines. And all of the odorant binding proteins in all the mosquito species had these 6 odorant binding proteins. And I think it was present in all the other organisms as well. What we interestingly figured out was this was not the only class of OBPs that were there. We identified new subfamilies which people had not seen earlier, such as the plus C OBPs. And we also coined this new subfamily, which we named atypical OBPs. And it is kind of adopted in the research environment right now, and people did a lot of analysis on the atypical OBPs from a wet lab perspective as well, where we found that there were 2 classic OBPs actually joining together to form these atypical odorant binding proteins. To the existing subfamilies that were there, we were able to actually add 2 main other subfamilies, which was minus C and 2 domain OBPs, or the atypical OBPs. So this was kind of a research that was quite well recognized. And I must say that the naming convention that we used in this paper has been adopted until today. Odor and binding proteins are named according to our paper that was published in 2012. And it has become as a reference paper for sequences odor and binding proteins in these three mosquito species. So we also went ahead and mapped these sequences on different chromosomes, and we figured out that subfamilies have preferred chromosomes, and these chromosomes also they were identical in the three different mosquito species and so on. And we characterized each of the subfamilies. We studied them in detail in phylogenetic trees, and we also were able to add value to the phylogenetic trees by identifying substrates to different subfamilies that are mapped on this thing. Probably I'll talk about it the next time when we discuss the structural aspects. So here, this is just sorry, this is just one such subfamily. So we did this for all the classic OBPs, minus C OBPs, plus C OBPs, and atypical OBPs. And we were able to actually characterize them into different subgroups, M type 1, MA type 2, 3, and 4. And interestingly, when we did ligand binding analysis, we were able to associate different odorant molecules that specifically bind to these different subfamilies as well. Yeah, thank you so much for the opportunity to talk about this work. It was a very exciting work, and I really enjoyed doing this work.
Yeah, thank you, Malini. Please stay on for questions that might come after this. But I now request Bhavika's recording to be played, and she's going to talk about her experience looking at honeybees or OBPs. And she has used evidence from starting from some of the Apis mellifera, but looked into Apis florea honeybees for the same odor and binding proteins. So this may be listened to and it's been recorded.
Hi everyone, my name is Bhavika Mam and I completed my PhD studying odor binding proteins with Professor R. Saudamini as my guide and mentor. As we know, insect OBPs are very important in olfaction and mediating chemosensation. Our study was published this month in Apidology, Springer Nature, where we identified 22 OBPs in eusocial honeybee Apis florea. And we further categorized them into classic and minus C subfamilies. Now, minus C OBPs are generally missing from bee genomes that are not honeybees. We used OBPs from the nearest relative of Apis florea, that is Apis mellifera, and we also used other public data to guide us in our study. In this process, we identified OBPs, few chemosensory proteins or CSPs, and a long non-coding RNA in Apis florea. Our study shows that minus C subfamily has evolved from the classic OBP subfamily in honeybees. In order to identify and analyze OBPs from Apis florea genome, we used a refined protein query set of Apis mellifera OBPs. We refined our query data set using 3 independent studies and a homology-based pipeline. Furthermore, we used publicly available RNA-seq exon data to refine our in silico predictions of gene models corresponding to OBPs. We annotated novel isoforms, corrected intron exon boundaries, and completed missing exons, coding RNA in Apis florea. Interestingly, CSPs have been involved in insecticide resistance, embryonic development, and regeneration. And this family of CSPs have been expanded in flower beetles and reduced in bees and wasps. Insect OBPs have characteristic conserved domains, conserved cysteine signatures, presence of signal peptide, and through our extensive workflow we talked about previously, we identified PBP GOBP domains in each putative OBP. We found that Cysteine signatures and interdisulfide spacing consequently in each subfamily across insect orders are conserved and comparable. For example, we can see here that a pair of classic OBPs in Anopheles gambiae and Apis florea have comparable cysteine signatures. A similar result is seen in -C subfamily OBPs from mosquito and honeybee. Next, we looked at locating signal peptides. A typical signal peptide has three regions, N, H and C, with conserved amino acid residues per region. The cleavage site determines the length of the mature signal peptide. Based on the physicochemical properties of a signal peptide, as shown in the previous slide, we wrote a rules-based algorithm written in-house that showed up to 70% accuracy on massive datasets such as signal peptide datasets from signal P and PREDICE. We also tried various other approaches and algorithms, including neural networks, blast tree alignment, hidden Markov models, and position weight matrix based methods. Phylogenetic inference revealed significant clustering of -C OBPs as a subclade of the classic OBP subfamily, comprising members of both Apis mellifera and Apis florea OBPs. OBPs of the -C subfamily, Apis florea OBP 14 to 20, they emerge closest to Apis florea OBP 13, which is a classic OBP with an observed 6 cysteine signature. To gain evolutionary perspective on insect OBPs, we constructed a phylogeny of insect OBPs from 11 orders from representative insect species. Interestingly, all the other classic OBPs clustered distinctly in a clade corresponding to the insect classic subfamily. However, Apis florea OBP 13 clusters closely with the -C group in a distinct subclade with high confidence. There is an increase in differentially expressed OBPs in response to insecticide and abiotic stress factors in both Apis cerana and Apis mellifera honeybees. This could possibly explain the positive selection of the C- subfamily. A similar co-clustering of classic and -C OBPs across other insects suggests a recurrent need for such variation in OBP subfamilies. Our comprehensive bioinformatic analysis suggests that -C OBPs may have most likely to have evolved from a classic OBP subfamily member in honeybees. It is possible that the evolution of such a subfamily could be an adaptation to the local niche of the insect species for functional specificity. Although classic OBPs in Apis florea are phylogenetically distant from -C OBPs, it is possible that -C OBPs in honeybees have evolved from a single ancestral classic OBP such as Apis florea OBP 13 of its species by deletion or mutation of the second and fifth cysteines.
Right. I hope you have enjoyed hearing this sub-story by Bhavika. What I wish to say is, starting to summarize this third story, what we have been hearing from Malini and later by Bhavika had been that the classic OBPs and the atypical OBPs are related because the atypical one seems to be twice as much as the classic ones. They seem to be harboring both of them. And whereas in the case of the -C or OBPs, they seem to have diverged from the classic OBPs as illustrated by Bhavika's computing efforts. And this was work done along with Snehal, because she had used, tweaked her Snehal's server to recognize the OBPs. So those are our three stories for today. We started off with bacterial chemosensors, some of the proteins that are involved in the generation of these chemicals that are participating in quantum sensing. And then we showed how they are habitat specific and they diverged accordingly. And then the second story was on the olfactory receptors And then the third story had been on the some of the insect odorant binding proteins. So I hope you found all these three stories very interesting. And also I hope we have been able to showcase the power of computing algorithms to recognize something very reliable and useful. And with this, I would like to thank the people who have been involved over time. And some of them have been for a long time in the, so for instance, the odor and binding proteins, the work really started sometime in 2006, and it is still going on, as you would have heard from Bhavika. And Rajas is the key person who had also narrated the success story, and Raghu Prasad Mejapalli, who had worked on the GPCRs. It really kind of laid foundation for our work on the olfactory receptors, which was performed by Naga and followed by Snehal, and then Vikas, who had actually taken up the web server and did benchmarking and so on. On the third story, we have different people involved. Malini, with whom you have heard the work on search for an analysis of mosquito OBPs, and then Snehal and Bhavika together had then very recently looked at some of the honeybee OBPs. So these are the people who had worked on these projects, not to mention the collaborators whose names I will mention then and there. Okay. So what I thought I will also share with you today is you have been hearing the third talk in our series. And in the first talk, we did discuss the protein sequence searches, right? And then that was my second PhD student, Saikat, and then the second PhD student, Anirban. And what I'm projected here are their current positions, where they are right now. And Sandhya, who's also been working on the cascade sequence searches. And the second story, we had talked about domains. And here delving deep into the protein domains, right? And here Chandrasekhar had come online, and I mentioned that he's working as a senior computational biologist in Cancer Research UK, and Divya, whose work on the Odours algorithm, which was narrated by Advait, who's currently with us. So Divya, however, is now in the Institute of Spice Research, and Anirban is back again. He was working on the phosphatases and therefore, but then Tina, who had applied it on non-mammalian genomes, is currently a postdoctoral fellow in Ireland. And in that story narrated, the three stories that have narrated today on the theme of olfactory proteins, Rajas, who had talked about the bacterial fluxes, he is currently a faculty in the Napoya Research Center. Raghu Prasad Medapalli, he's in the Casing Clinic in the US, and Snehal is currently a scientist in a company, Exelra, and Malini, whom we heard, she's currently a founder, co-founder, and CEO in a Deepnomics company. And then we have heard a bit of the InsectOR webserver from Vikas, who's currently with us. And Naga also, you had heard on the DOR database, she's currently with us. Whereas Bhavika is currently a postdoctoral fellow in Stanford University. So these are some group pictures that I would like to flash across before we take questions. But before this, I would request Shakti to share some of the papers that talk in detail about the works and the three stories that we had shared today. So Shakti, please, can you share the publications, so that people can go through in detail, whoever is interested. So I would like to thank the audience for being with us. And I see a few people like Dr. Alexander Breven, who's been listening to all the three talks so far. So please do stay tuned. So Shakti, please.
Thank you, ma'am. Here, what we have listed the most cited research from the short stories what you heard today. So first, Lexus proteins Rajesh presented, and GPCR Raghu Prasad Rao's work ma'am presented today, the two papers. And we heard about ORs. There are two papers from Snehal et al. And one paper about DOR database from Nagaratnam. And we have in alternate binding proteins, we have Malini's work regarding alternate binding proteins in mosquitoes. And also we have a new paper from Bhavika regarding alternate binding proteins in honeybees. Yeah, and what I wanted to tell next, we have next week new sets of stories coming up. We have titled it Being to Few Protein Cures. So these stories includes serine proteases, proline oligopeptidases, and GPCRs. Please stay tuned.
Okay, thank you. Thank you, Shafi. So I go back to my sharing of the screen. So what I want to project are some of the photographs, because this is sharing the history, right, over many years. And I hope you have enjoyed the 3 stories today, and some pictures that I would like to share. With this, I think we are all set, ready to take any questions, and we can see some that have come in the chat box. So let's look at them, shall we? So I would request people like Vikas, Manini, and Rajas particularly to come online, meaning with the video on, so that we can take questions as the case may be. And we have a few questions here in front of us. The first question is to Rajas. The question is, how are the autoinducer 1 and 2 different?
So autoinducer 1 is used for species-specific communication. So different bacteria, a particular species of bacteria uses autoinducer 1 to communicate with other members of the same species, while autoinducer 2 is common between different bacterial species. So different bacteria in the same ecosystem or habitat use autoinducer 2 to communicate among themselves.
Thanks, Rajas. The next question is, how did you look at the phylogeny from the structural point of view for so many sequences? Was there any specific tool used to do that?
Yeah, so we chose a representative from each of the cluster we got from the phylogenetic tree. So we used certain criteria to arrive into the candidate sequences. For instance, sequence identity with the already solved Luxus structures, and for instance, the level of annotation of the protein in the UniProt database. So these were some of the factors which we looked into while zeroing into the representatives, from which we constructed the 3D homology models. Once we got the 3D homology models, we performed structure-based sequence alignment of all the homology models, and that's where we compared divergences in the 3D coordinates of different of the Luxus residents.
Thank you, Rajas. We have another question just by Aditi Patak. So she asks, does the OR algorithm account for introns and alternate splicing events? So perhaps Vikas would like to take this question.
Yes, so for initial alignment of the known query sequences to the genome, it does check for the intron length. We can set the maximum intron length also as per the insect species. And also it checks for the alternate splicing sites while building the gene model. So we finally consider those gene models which are scoring better, considering all the possible alternate splicing sites.
All right. Okay, so we have another question. It goes like this. While using the database, can we set filters like coverage manually? In case if the alignment is shown at 2 positions, how does it deal with chimeric alignment?
Yeah, okay, so we can change the completeness or the coverage criteria manually, and for each different species we want to. So by default, it is 300 amino acid based on the analysis of the OVA sequences which we had done. And the next part was okay for the chimeric alignment. Yeah, so in the chimeric alignment, if we have the one part of the alignment is coming among the best query for that particular subcluster, then we'll consider the fragments of that alignment Otherwise we will discard that alignment.
Okay. There is one more question by Pankaj. It goes like this: is it possible to make a DL model for OR algorithm?
Yes, definitely we can make that. It's just that we need a very good or properly annotated many OR sequences, which I think are not in abundance. Efforts are going on to sequence many more insect species and annotate them properly so that we can have a good data set.
Let me check if there are any more questions. Yes. There is another question. What was the basis for selection of organisms for DOR? Perhaps I can take this question. Yeah, so the DOR was like the beginning. So we wanted to pick up representative model organisms that have gone through a relatively higher amount of annotations. So that's why we also wanted to take these species and the genomes of the species such that they would span and represent different evolutionary time scales. That's why we had chosen Rostrilla, C. elegans, mouse, and of course human, because it finally has to apply to humans. Otherwise, we wouldn't be so interested in analysis. Yeah. So that's why these different organisms were chosen for DOR. Thank you. And then there is another question. It is by Sajeevan. It says, thanks for the great talks and the history of Lab 25. Thank you, Sajeevan. And then he goes on to ask, over curiosity, do plants have similar OR sort of genes? Plant can sense, smell, and respond. Any information known? Well, it's an open question because I think they perhaps Shakti would like to take. No, I think what I could imagine is that the plants seem to operate by means of protein-protein interactions, and their mechanism of transduction is driven by hormonal signaling and so on. Yeah. Having said that, we've not chosen Arabidopsis, so I think the plant biologists will be cross that we've not done that. So we can look up for olfactory signal machinery in plants. Thanks for the very nice question. Okay, any more questions coming along? Okay, so I think we have taken questions that people have put up in the chat box so far. And if anyone else would like to ask a question, a burning question that you just like to pick up now, you can do so. You can raise your hand, and then we will try to bring you perhaps into the post panelist channel, and you can ask your question, or you can type your question. So there is a ready for all that. But even as we are speaking, we have now 1 hour 17 minutes on this seminar, and if there are no more questions on the chat box and no hands that get raised, so we would view this as the end of the session. So I think we had a great fun sharing our stories, which have spanned from 2002 until 2023. Yeah, so that's the philology paper. So this has gone through a huge broad time scale, and we have chosen this theme to only talk about the olfactory proteins that we have chosen for a computational exercise. I hope you had enjoyed the session as much as we did, and thanks to the audience for your patience, and thanks to the help that we have been receiving from the communications team at NCBS for being able to host this session. And I also thank the volunteers, and also thanks to the audience for the very nice questions. And please stay tuned. We will see next week at the same time, 3 p.m. Indian time, next Thursday, on the topic that Shakti has already mentioned. Goodbye. Stay safe. Bye.